Hello, in this video we're going to be looking at the processes involved in Meiosis II. So let's not waste any of your time. Let's get right to it. Okay, now when we're talking Meiosis II, one of the things we want to remind ourselves is that the stage names, the phase names, hey, they're the same, except now we have a reference to number 2 behind each of the words. The stages are the same, just like mitosis, just like meiosis 1, except now we're going to see a number 2 behind them. We also want to remind ourselves that, and you can see in the diagram beside me, the 2 red sister chromatids, but then the purple individual chromosomes. Those are referencing the same thing, but what we want to remind ourselves about is that when we see a reference to the words sister chromatids, we're talking about meiosis II, because we're going to get down to individual chromosomes, no longer in pairs. No longer 2 sets of pairs, but down to the individual sister chromatids. Now in meiosis II, again, here's a reference to those words. The idea of meiosis II, we're looking at we start off with those 2 cells, and we go through prophase 2, metaphase 2, anaphase 2, and telophase 2. And when we do that, now we're going to get down to 4 haploid gamete cells. That's the final product of meiosis II. Now the path to get there obviously starts with prophase 2. After cytokinesis of meiosis 1, there's a brief interphase, do a couple of checkpoints, make sure things are going well, but no duplication. Okay, we're not doing that. That would just start the process over again. Okay, again, no S phase, and now we've got the chromosomes ready to go. They're going to get moved to the middle. They're going to get separated as sister chromatids, and then we're going to divide that cell up. Now, and here in metaphase 2, this diagram, you may be thinking, what's going on here? Because it's a picture of them lining up, but the pictures aren't the same. The picture above is a reminder of what it looked like in metaphase 1, and the bottom is metaphase 2. So you can see in metaphase 1 we had the homologous pairs of chromosomes, but now we just have the 1 chromosome with the sister chromatids. But again, those spindle fibers, moving them to the middle, and of course they're attached, and eventually will snap and pull those sister chromatids towards the poles. So now we're in anaphase 2, and again the picture at the top is a reminder of what it looked like in anaphase 1, and the picture at the bottom is anaphase 2. That microtubule pulls them to the poles, and now we're just separating the sister chromatids apart, and you can still see the effects of crossover down there. So that's just going to emphasize that we are really blending the alleles and the traits up to create as much variation in the offspring as we can. You can see that in the diagram at the bottom in the anaphase 2 example, the sister chromatids are going to the top. You can see the one on the far above the other. It's not really left, but it's above. it's mostly blue with a little bit of red, and then the vice versa, a lot of red, a little bit of the blue. And of course, the two at the bottom, they are all red and all blue. They didn't have any, they don't show any signs of crossover. So it's really going to, when you think about those 4 chromosomes, when they get separated into individual gamete cells, that really creates some variation. Any one of those could make a very similar offspring to what the parent was, or you can have a lots of variation in that. Alright, so now here we are at telophase 2. Again, the picture at the top is telophase 1, picture at the bottom is telophase 2. And now we're going to separate again, okay, those chromosomes, and again we're going to show 1 going into 2, but remember there's 2 going into 4 for that final product. And remember, the offspring, no 2 cells are going to be alike. Okay, we've gone through crossover, we've gone through independent assortment, and so we've really mixed up those alleles to some degree to try to create as much variation as possible. Okay, so just again a reminder of what we've got going on here. We started off with the 1 cell. We went through meiosis 1 to get 2, and meiosis 2 to get 4, with as much variation as possible. 4 haploid cells, meaning 1 set from each parent. Oh, I got my mouse too low on my browser bar. Alright, so again, just another reference to this whole process of going from 1 to 2 after round 1, and then to 4 after round 2. Well, that's our video over meiosis 2. When we come back next time, we're going to be looking at things that can go wrong. In other words, errors. So we'll see you then.